So I'm the scientific founder and chief scientific officer at Hemologics, and I've got more than 25 years of experience in the fields of immunology, hematology, translational medicine, and drug development. My PhD project involved isolating the DNA sequences of the original monoclonal antibody that was the precursor to Capimab. And subsequently, I was involved in genetically engineering our lead therapeutic antibody for the treatment of multiple myeloma. I've been involved in taking this antibody from laboratory through to preclinical development and now in clinical development. I was also involved in the design of the KMA CAR-T construct in collaboration with my colleagues at the Westmead Institute of Medical Research. And subsequently, I lead the company's team in the preclinical development and translational studies and clinical development program, which is being conducted at the Peter MacCallum Cancer Centre and Centre of Excellence in Melbourne. We've also been doing preclinical research and translational studies with our lambda antibodies, the LMA CAR T cell, and we have bispecific antibodies in early development. The study showed that KMA and LMA are present on malignant plasma cells, and most importantly, they're not present on normal plasma cells. We looked at bone marrow samples from a range of diseases, including multiple myeloma and its precursor diseases, which are known as monoclonal gammopathy of unknown significance, and also smoldering multiple myeloma. And we also looked at plasmacytomas and another disease that's known as light chain amyloidosis. And all of these diseases are commonly known as plasma cell dyscrasias. And we found that KMA and LMA across all of these stages of disease are only present on malignant cells and not on normal plasma cells. I think the data is important because it showed that these diseases can be separated into either KMA positive or LMA positive malignant cells. And we're developing our monoclonal antibodies, our CAR T cells, and bispecific antibodies that are very specific to either KMA or LMA targets, which allows our immunotherapies to only destroy the disease cell and not the normal immune cells. So essentially, this is precision medicine because we can identify patients who are potentially going to benefit from a highly specific and targeted treatment, and at the same time, preserve their normal immune system. The data from previous studies has shown that KMA is not on plasma cells in normal bone marrow samples or on blood smears. And this study confirmed that LMA is also unique to the malignant plasma cells and not found on normal plasma cells. This means that our immunotherapies will only destroy the malignant plasma cells and leave the normal bone marrow plasma cells intact so that they can produce antibodies or immunoglobulins against bacterial or viral infections, and importantly, retain vaccine responsiveness, especially with the universal sort of COVID-19 infections. In general, other immunotherapies target proteins that are found on normal plasma cells, as well as the malignant plasma cells. For example, B-cell maturation antigen, which is commonly abbreviated to BCMA. Another example is GPRC5D. Now, these are found on the malignant cells, but they're also found on normal plasma cells, normal plasmablasts, and normal B cells. So the depletion of normal plasma cells results in the loss of serum immunoglobulins. And these patients who are treated with these immunotherapies become hypergammaglobulinic, which means that they require infusions of normal gamma globulins because they've lost their normal plasma cells, so they cannot produce antibodies against infections. So they are prone to infections, which can be life-threatening, and their immune systems are not responsive to vaccines. Our lead antibody is called Capimab. It was formerly called MDX1097 in our previous publications. It's been tested in 3 clinical trials in myeloma patients who were previously treated, and all 3 studies confirmed that the antibody is very well tolerated. But most importantly, there were no antibody-related hematological or immune cell toxicities. And specifically, there were no lymphopenias, which means there was no loss of B-cell lymphocytes. A phase 2b clinical trial of Capimab in combination with a typical standard of care, which is Revlimid and dexamethasone, was conducted in patients who'd previously been on treatment, but their disease was progressing. We observed an overall response rate of 83% versus 45% in patients who were treated with Revlimid and dexamethasone alone. And this was a contemporaneous matched control group. There was also a 46% reduction in the risk of death versus the matched controls. So we've not only seen that the antibody has a very good safety profile in the treatment of myeloma, but is also efficacious and can be used in combination with the current standard of treatment. Yeah, this recent publication and our clinical trials have shown that our immunotherapies can potentially target and destroy the malignant cells and leave the normal cells intact, especially normal immune cells. This is true not only for multiple myeloma, but also for the precursor stages of myeloma disease, such as monoclonal gammopathy of unknown significance and smoldering multiple myeloma. In general, multiple myeloma stems from smoldering multiple myeloma. And the safety of our monoclonal antibodies suggests that they could be used in earlier stages of the disease before it actually causes organ damage in the patients. The preclinical and in vivo data was compelling. And this is the reason we're going into the phase 1 clinical trial. The data showed that the KMA CAR T cell has retained the specificity of Capimab, and it only destroys the malignant cells that express KMA. And it doesn't, we anticipate that in these myeloma patients, the normal plasma cells will not be depleted, very much like the monoclonal antibody. Potentially, this would mean that a patient would have a normal immune response to bacterial and viral infections and perhaps a better quality of life during and post-treatment, because they won't be subjected to these life-threatening infections, which means that they have to isolate away from family and away essentially from the community so that they do not get these infections. So we anticipate that they will have a better quality of life as well. Well, we know that more patients are being treated with BCMA immunotherapies, and this means that those who relapse from antigen escape or from other reasons why the immunotherapy stops working, there will be very few treatment options. KMA and LMA studies showed that our targets actually increase in density on the surface of the myeloma cell in treated patients, and therefore they offer a new target and a new treatment paradigm for patients who have failed on current standard of care. And in some cases of plasma cell dyscrasia, we actually showed that KMA and LMA are present on the malignant cell, but BCMA was not. And this means that we have the potential to treat patients whose malignant cells don't express BCMA, or those who have suffered from BCMA loss due to the selection pressure of the current immunotherapies. So we potentially do offer a paradigm shift to patients who have been treated with BCMA treatments and their disease has relapsed, but also at an earlier stage of the disease, in particular for monoclonal gammopathy of unknown significance and also smoldering multiple myeloma, because we have shown such an excellent safety profile in the clinic. And we do know that our targets are not present on the normal plasma cells in the bone marrow, and certainly not on B cells in the blood.